Oh, I had an energy this morning. Did you? Where you live? In this one. And they're hiring new people. No, I would be This is kind of a small practice. So they opened this clinic where it's not enough life balance. Okay. Good. Oh, yeah.
Start this. I started it.
It's horrible. Oh my gosh.
Oh yeah.
It was so bad. Bro. And it's like the $4. They walking across oncoming traffic. This area is scary. I don't like walking out here at night. People live a couple of spots like this now. Yeah. How do you go? It was like literally, I was in Congress for like 40 minutes. Oh yeah, it's coming from this. But if I like, yeah, because everyone's trying to take back roads. And it's like the cheap air up there, so everyone. I live in there here. And I get sick on rides. Also there a couple of years ago. Because Aaron Morrison was playing. And I was going to go school out in the car because that one I was like, it's only an hour. Might as well. Also, of course. I like the Broken Arrow area. Yeah. Where it's kind of quiet. Yeah. But the Tulsa downtown. Yeah. Tulsa is a bit sketchy, especially if you don't know where you're driving. Oh, yeah. I went to go get hay for my horses one time. And I was trying to stop at a gas station and I needed to go to the bathroom. And I got lost. And I called my mom and I was like, I'm lost. And she came over to my dad because he used to live in Tulsa, so he knows the area really well. And he was like, what are you doing there? Like, yeah. He was like, yeah. Yes, because of like one leg and stuff, and then some I'm not like where I was. Yeah. If you really want to go to a sketchy place in the city, go to the capital area. I went there for a conference one time, and I yeah, okay, so state capital area, like where the Capitol building is. Sketchiest place in the world. Yeah, the marathon. Yeah. Yeah, my friend was trying to run one person. It was he lived over there, and it was like pitch black, and he was standing over there. Like 6 a.m.
I don't know. That's what I was thinking. I was like, are we gonna have any more people come? Or not? I guess not. I mean, we're at start time, so it's whatever. Just to be clear, I grew up in Tulsa, so I'm just letting it go. I think Tulsa is better than Oklahoma City. Yeah, by far. Yeah. So I think that's what it is. You know it, and I mean, I know both now because I've been here for a really long time. But now, when I look back and I know the areas of both what's safe and what's not, I think Tulsa is just more liberal. It leans more liberal than Oklahoma City. I think that's why I like it a little bit more. But
Oklahoma City's getting to be like that.
it is. They're gentrifying everything. It is a lot, especially if you're going down like 23rd or by the Paseo and all of that.
And then you go to the rural area.
Yeah, it's like, well, never mind. We're still in Oklahoma after all.
I used to live in out west.
Mhm. Yeah, I like it over there. I used to like do a lot of my shopping at the Asian market over there, the Townwind place.
About 2 hours. I work in Arena.
Yeah, that's.
So,
We'll go ahead and get started. We don't have a Kahoot today, obviously, because we didn't have, that was last time. So we'll start back up with basically where we started. And hopefully we'll have time. I know you guys are just gonna love it, but hopefully we'll have time for questions today. And at least one set of them, but it's a pretty heavy chapter. So it's a lot. It's just a lot. And it's at least it's like one of those chapters where I feel like we'll cover it and like the basics of it are really all you have to take with you. I feel like for the next one, of course, you have to know all the details for the exam, but you know, it's just a lot. So we'll see what I mean. So, right. So we have talked a little bit about genetics. I introduced it, and this is the whole reason why I introduced it, is so that when we got into this chapter, it wouldn't be like a big jump, right? So, and we'll have, you know, DNA. And we were talking about how DNA is basically like we keep it in our nucleus. So you'll have to make a copy of it, which is mRNA, a working copy. And that can be turned into protein, right? We're going to go through all of these steps today, which is why I'm writing on the board so we can refer back to the basics when we need to remember where we are and what's what, right? So DNA becoming RNA. That's basically like our language here. We're speaking in still nucleic acid. So our language here is going to be nucleic acid. That's definitely an E. All right, and then the language down here, though, that's a different language, right? So that language and here we're literally talking about the monomers, the things that we were saying make up DNA of RNA. What is it? Nucleotides, right? With our nucleic acid. It literally says NA, nucleic acid. What its language is speaking is right there. Proteins, just you're going to have to remember, proteins are made out of amino acids linked together, right? So our language here will be the amino acid. These are both speaking in nucleic acid, and now we're shifting to a different language here. Amino acid. So we'll call this transcription. The script you would think of writing or like a scribe that somebody who writes. You're generally going to be speaking the same language as we're doing that, like when you guys are taking notes, right? You can think of my slides as being basically the DNA and the little notes that you're taking for it to help you move up as being the RNA. That's the working copy of it. So that's transcription. It's all in the same language. But however, now we need to turn this into proteins, a different language. So just like we would with actual language, we're going to translate it. This is called translation. What that means as far as like actual language goes, and this is exactly what's going on here. We're changing the language now. We have to translate it from nucleic acid into amino acid. But that's the gist of what we're going to go into. Another thing we're going to talk about, what we're going to talk about first, is going to be DNA and making copies of that DNA. That process here, going from DNA to making copies, is just called, nothing fancy, replication. We're just replicating, and that's all that's happening. Okay? So you can see the difference in these words that we're now talking about. Replication, replicating the DNA. Transcription, we are rewriting it into our working form, but it's transcription, right? Taking that message, the only message that we want in it. that one gene out of the 20,000 genes that we have, that one gene taking it into that working format. Okay? That's the RNA, that's the transcription. And then taking that and making protein out of it is the translation. So these are the big terms that we'll need to be familiar with for this chapter. There's other terms, of course, but I feel like when we get on to describing some of these steps, whether it's this chapter or future chapters, knowing these helps a lot. So, all right. Now we get the general idea of it. All of the genetic coding material in your cell, whether that is what's in your chromosomes, literally, or what is carried in any extra information, extraneous information. If it's encoded genetically, anything in that cell, if it is included as your genome. Okay, and what I'm trying to get at is here, bacteria can have plasmids. That's little extra bits of DNA. They're not necessary for survival, but can help with survival. If they have a plasmid, that's part of what's going on genetically inside of the cell. It's not part of their chromosome, though. It's just part of their genome, right? Not part of the actual chromosomal structure, but it is part of the genome. Another good example, remember how we had said how mitochondria have their own DNA, their own circular chromosomes, and they'll replicate on their own and everything like that, right? So our chromosomes don't include that in their information. It's a separate thing, but it's still included in our genome, right? That's what that's talking about. Remember that cells, if it's a cell, it has to have DNA as the genome. If you are a virus, you can have DNA or RNA. It's basically just skipping that step of transcription. Alrighty, and you know, we talked about a virus, at least one, that can go back and forth, which is kind of not helpful for understanding the process. All right, so a chromosome is just a single unit that would be packaging that into. Bacteria always have one, I say always, typically have one big circle as their chromosome, just one, and it's circular, typically. Our chromosomes, we've kind of talked about them before. Your chromosome might exist as this. You have one from the mom and one from the dad. Mother and father, right? And then whenever you're going through mitosis, you'll have to copy these so that you can be sure that the new cells have the right amount of those, right? So we'll go from this, and this cell will then, after it's replicated, the DNA, right? We're gonna go through replication. It looks like X's to us. This is where it's gonna look like X's. And then they'll line up and we'll pull them apart. Remember that when we went through mitosis and PMAT and all of that, okay? So then the new cells will each have MF and MF. This is a little bit important for what we're gonna get into in a little while, which is actual genetics. Not just talking about DNA and what it does, but actual genetics. We're not going to get too much into it, no worries, but we're going to talk about it a little bit. There's only 3 kinds of genes in the known genomes. It's going to be either genes that code for proteins, genes that code for the RNA machinery that's going to be used for the proteins. That'll be the ribosomal RNA and the transfer RNA, which we'll talk about. And then the regulatory genes. These are genes that are involved in controlling whether those proteins are going to be made or not, whether these genes are turned on or turned off. Now, if I tell you that every single gene in your whole genome that makes you up is in every single cell of your body, right? You have all that information in every cell of your body. So why do you have liver cells versus skin cells versus intestines? Versus how do they know? So that's all going to be regulated by these regulatory genes. So they all have the ability to be liver cells. They all have the ability to be intestinal cells or skin cells or whatever it is. But they're being regulated to make those specific tissues. It starts early on, of course, in embryonic development. They're getting signals to make certain kinds of cells when they're going through that. And then once you've been kind of signaled to be that, they can't really go back. That's why we have stem cells versus like typical cells. But that's it. The regulatory genes are what's going to be turned on and off, even though you have all of those genes in all of your cells. All right. This is it. This is the only thing we're going to be talking about with actual genetics. Okay, so I promise. I didn't ever plug in my. So, genotype versus phenotype. Plug in this thing. We're going to do a Punnett square today. I don't know if you've heard of it, but you'll probably have seen one before. All right, imagine for me that you have some genes that code for different things in your body. Wow, go figure that, right? Let's say one of those genes is for your hair color. So let's say big B will represent having brown hair. I'll just write it up here, brown, so you know. And little b, which is just another version of that same gene, we're just going to say that one's blonde. Typically, when you see geneticists writing them like this with big and little, it's talking about whether something is dominant or recessive, typically. I'm not going to necessarily test you guys on that, but it helps, I think, to understand what's going on whenever we have babies and stuff, okay? So if you had a gene, let's say your mom, okay? This is your mom's genetic makeup in her cells from her mother and her father makeup. She had carried one big B and one little b. Your mother came from, this is grandma and grandpa. Grandpa donated that little b, grandma donated the big B. Okay, we could go back and back and back and say where these are all coming from, but it's not important, okay? Because every person has two copies of whatever. All right, then let's say your father, let's just say he has the same genetic makeup. Let's get rid of this because we already know what this means now. Your father, let's get out of here, man. Get out. Has a big B and a little b also. This might be from his mom. So this will be grandma on dad's side, and this will be grandpa on dad's side. Okay, so this is the father and the mother, and they have gametes, right? That's what we call them officially, but sperm and egg that are going to come together. And we're going to talk about what their genetic makeup is. So if mom has big B and little b carried in her genes, so that means she's carrying a gene for brown hair and a gene for blonde hair. What that means. She's got one from each. The big B is dominant. And what that means is that your body has signals that tells it to express that in favor of the other thing. I'm sure nobody in here is going to argue that brown hair would be dominant because it is easily the most common hair color that there is. So, so let's say, for sake of argument, that blonde would be recessive to brown, right? So you're carrying that gene in the background, but you're not expressing it because this is the one that's going to be expressed. And this is true for a lot of your genes. You're typically going to have two, one from your mom and one from your dad, but you're only going to be expressing one. Your body doesn't need both of them expressed at any time. It only needs one. So how does it know which one? Well, it will know based on dominance or recessiveness and by other signals that might be going on in the cell, favoring one or the other. And depending on the cell, right? So that's another thing. But so it gets real complicated, but as far as making the new person goes, it's not too complicated. So you can give your genes in the following way. You will, let's say we're moving dad's genes down and mom's genes across. Her big B, as it goes across, big B, it'll go here. Her little b, as it goes across, little b, little b. All right, so if we move dad's now, big B will go here, big B will go here, and then his little b will go here, and little b will go here. So if we were remembering that big B means brown, and that it is dominant over being blonde, 75% chance that the child will be brown hair. Now that's what they're going to show, right? As what they're displaying, not their genes. In their genes, they might be hiding that little b. But what they'll actually show will be brown hair. This guy down here has to have the two recessives so that the body has no other choice but to express that gene. That's the only time you get recessives, true recessives anyway, to be expressed. You have a 25% chance that the child would be blonde. This is in every single time that you have a meeting of the sperm and egg between these people. Every time. Not like you have 4 kids and so 25% of them will have blonde hair. That we go back to square one every time we have a meeting of a sperm and an egg. So just because you already had one blonde hair kid doesn't mean that you're in favor necessarily. Or not. That's not how it's determined. So this is the possibilities of what you can see. You can see why dominant traits are going to then be expressed more often and seen more often, because they're going to be basically kind of silencing out the recessive ones. It's important to understand, because if we're not talking about hair, if we're not talking about, eye color or something like that, which is just sort of aesthetics, if we're talking about whether or not you can, make certain proteins in your body that help you live appropriately, that might lead to having deformities, that might lead you to being able to survive past, being born, things like that, then it becomes a little bit more serious what we're talking about here with dominant versus recessive. So, and it also matters like what chromosome we're talking on. In the future, we're going to learn about X-linked SCID, which is severe combined immunodeficiency. And X-linked SCID means that it's linked to genes on the X chromosome. Go figure. So I don't know if you guys know this. I'm sure you do. This is just me being facetious, okay? Women, if you're female, genetically speaking, you will have two X's, right? And the male, genetically speaking, will have an X and a Y on the sex chromosomes, right? You have two of everything, and you had to get one from the mom, and all she had was X's to give, right? So you, as a male, you just got a Y from the dad. So this is our options. If you had a gene that was right here, and it was bad, somehow you had a mutation, you can't make this protein or whatever, odds are you're probably going to be able to make it on the other one. What happens if you only have one of these though? The Y does not carry the same genes. So you're stuck with that one X. So if it's messed up on that one X, there's no other gene to cover it. And that's exactly what happens with a lot of X-linked diseases like SCID. SCID is very uncommon, and there's a reason why, but it's severe, and when I say severe, I mean it's severe. Typically, X-linked SCID is going to be the one where you have no immune system at all. You don't make antibodies, you don't have innate immunity, you don't have T cells, nothing. You have nothing. And so they are the boy in the bubble. They're the boy in the bubble because it only happens on males, because they only have one X. And typically, if they're carrying that bad gene as a male, they're not going to reproduce and spread that on, because typically they're going to die before they ever get a chance to reproduce. Most of the time they die when they're a kid. Like unlikely that they'll make it past being a teenager, no matter what we do. Just at some point, some minor thing will cause an infection. We have no idea, really no idea how many things you're up against every single day. When you go outside, when you get in bed, when you let your dog lick you, when you hug your kids, when you eat your food, how much bacteria you're introducing into your body that your body can kick out because it's seen it before, it's dealt with it, it's no big deal. But that person can't ever, no matter how many times they're exposed to it. They can't get vaccines, doesn't matter, it doesn't work for them because they don't have antibodies. They can't make them. So that's terrifying to think about. It's pretty uncommon. There are different levels of SCID. There are some that are not X-linked, that are linked to other genes, but they still consider it falling into this category. So females can get that, but typically you're almost never going to see a female get it because they'd have to have two bad versions of the gene. And so you have to get one of those bad versions from a father. That's just not going to happen. So the mother will be carrying it secretly and hiding. And then when she has a boy, she's passing on that trait to the child. It tends to be, as I'm sure you can imagine, would tend to be mutation based, not really like hereditary, if that makes sense. So, okay. But this is why I bring it up, because you guys, most of you guys are going into medicine. If you're going into vet, genetics still applies to animals, and some of this stuff will actually be coming up in animals if you were treating animals, as far as like how it can affect certain, like why this German Shepherd is all white versus having color. But that can lead to, if they're all white and they're all recessive because of that being expressed, what other genes do they have that are recessive that can make them more likely? And why is it an issue when we think about pets and we're buying our signed off AKC registered animal? Why that's a problem? The problem, not necessarily because of the breeders and how they treat them, which is what a lot of people would think about. Even if they're being humanely treated while they're being bred, These dogs are severely inbred, severely inbred. And that's why we have the different breeds that we do. But like you'll look at something like a bulldog or a pug. And bulldogs are like last line gen. They can't be, you can't get any more animals of different breeds off of them because they've been so messed up that they can't be, bred out to anything else. And you guys know, I'm sure, as you can imagine, they have horrible breathing problems. And a lot of these animals having, just bad hips. Look at, German Shepherds, we think of them as being, active dogs working for the, police and whatever, but there's a reason they don't stay in the police for very long because they get hip dysplasia and they can't anymore. They just can't run or jump or anything. And that's not because that's just how dogs are. That's because they were inbred. So inbreeding literally means you're going to be taking somebody that is like XB and then, the big B, little b, and big B, little b, and you get the little b. And then if they're getting up with their cousin or their brother or whatever it is, which is what happens in dogs and where they originated from, then you're going to be just way more likely to express that little b, little b. It's going to increase in frequency more and more and more in that population. And that's exactly what we see in dogs. You're choosing specific traits that way. You get certain traits that are good, but then unfortunately you get the bad with them. You don't get to choose. So that's how it goes. That's why I want to bring it up. This is one of those things that may even apply more to veterinary than it does to, depending on what field you're going into in nursing. If you're looking into things like cancer, you will be dealing with genetics like nobody's business because a lot of this is genetically related. But anyways, so that's that. So there's the genotype. So big B, little b, that would be your genotype. That's the genes you're carrying. The big B from the mom and the little b from the dad. Great. Your phenotype is what you're expressing. So you are big B, little b, you have brown hair. Can you tell by looking at somebody with brown hair whether they are big B, little b, or big B, big B? No, right? You can't tell just by looking at the fact they have brown hair. Everybody, if they had brown hair, big B, or big B, little b. Having just one B is enough. Big B, is enough. So yeah, you could maybe tell if you looked at somebody who's blonde, in this case, right? You would know. If they are blonde, they have to be little b, little b. You would know it. So some things you can tell and some things you can't. But phenotype is referring to what you can see, what's expressed, and genotype is what the genes say in the background. All righty. So we've seen this about DNA and nucleotides. Nucleotides are going to be a sugar with a phosphate with a nitrogenous base. So if we're talking about DNA, that sugar is deoxyribose. All right. And that nitrogenous base is going to be A, T, C, or G. I'm not going to try to trip you up with these names whenever we take the test, but be able to recognize that adenine is, in fact, a nitrogenous base, right? So the A, T, C, G, when you guys hear me say that, because that's pretty much what I'll say, know that that means adenine, thymine, cytosine, and guanine. And if I say later on cytosine, blah, 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 you know what that means. Okay, so just be familiar with these terms. A always pairs with T and C always pairs with G in the genetic code. What do I mean by pairs? We're talking about the different sides of the double helix. When you unwind that double helix and all you're left with is a ladder, if this side had an A, the other side has to have a T. If this side has a C, the other side has to have a G, and so on and so forth. Okay, so that's how DNA is going to be like structured. So that's where the nitrogenous bases are in all of this. They're there. This isn't the whole, this right here isn't the whole nucleotide. That's just the nitrogenous base. All right. Back here, right here, we would have the sugar and it would be bound to a phosphate, a PO4, whatever. And then sugar, phosphate, sugar, phosphate. Your nitrogenous base, so I'll just put N base because I don't want to write it out. Your nitrogenous base is always in DNA or any other nucleic acid anyways, always bound to the sugar, not the phosphate. Okay, so the whole unit, phosphate with sugar with nitrogenous base, that's one nucleotide, and they're going to be bound together over and over and over again. this is going to get ridiculous here in a minute as far as how that's going to be oriented and everything, because we're going to talk about anti-parallel arrangement. Okay, one side of our double-sided helix runs in one direction and the other runs in the other direction. Isn't that nice to imagine how that makes no sense to anybody in their mind? So that's okay. That's what I'm here for, right? So we had our ladder and we already talked about how it's set up and how these guys are going to be complementary across from one another. A, C, C, G, T, A. Or you could write Gattaca if you know that movie. This is T, G, back before Ethan Hawke. Ethan Hawke used to play like heroes. Now he plays like creepy guys. C, A, probably don't even know who I'm talking about. It's okay. Yeah, it's a good one. So we have our little things set up. We can see that the things are lining up and we're binding with what we should be. And now I'm going to tell you that these backbones here, this line along the whole back, they go in opposite directions. So this one, we would call it the 5' end. And this is the 3'. I'm going to show you what that means here in a second. Just hang in there. I just can't draw the chemical structures worth a damn. It would just look like nonsense if I tried. So, and then this one will go from 5' to 3'. So they're going in opposite directions. All right, the backbones are. And then this one and this way. Oh, that's really good. Okay, so that's the orientation you can think of. I will almost always be referring to things moving in the 5' to 3'. And yes, this is going to carry on, and I hate to break it to you. But 5' to 3', that is an important direction to remember. All right. The other side is going in the opposite direction. So it's kind of opposite in a couple different ways, right? We have the complementary sequence, it's kind of the opposite, and now the direction is also opposite. So we call this anti-parallel because they're running parallel, but in different directions. So anti-parallel. It's hugely important in this. So when we look at the actual chemical structure, this is why I couldn't draw it. Y'all do not want to see it. I remember when I was in biochem, and we were taking a test over this. We had to be able to like recognize the entire structure. And sometimes we had to actually draw chemical structures. Y'all, I got so carried away with it one night. I started just drawing like whole like DNA strands, and I had like papers and papers like out on my bed, like trying to draw the DNA, but you can't quite get it right because of the angle of the, anyways, just don't be that. Okay. You don't need to be that. That's me and what it looks like when I'm obsessing over a thing. So anyway, so we said 5' to 3', right? That's the direction that we would want to be going. And the other one is just oriented opposite wise. What that means is, if you guys remember learning in chemistry, how we number our carbons on things, like especially on like sugars or something, our organic molecules. Here, we're going to be numbering these carbons. So this is 1, 2, 3, 4, and 5, or whatever. I don't really care what you want to call them. So one of these is an oxygen in there. But anyways, based on how that sugar bonding setup is, whatever carbon is at that end is what we call it. So in this case, we have the 5' hanging out, so whatever the number 5 carbon hanging out from your sugar on this end, and the number 3 carbon hanging out. And you can see it when you look at it this way. If you're just looking at these segments of the sugars, you can see where the 3 is at the end and not the 5 versus the other way at the top. Yeah. So that's what that's talking about. That's all it means. And I will not make you draw it. I will not make you point out which carbon is which ever. But that's what it means when I say that, so that you know that it's not just some random number that you don't understand what it is. It came from a thing. It came from that sugar and numbering it. And you can see here that the 3' up here and the 5' down here, that's where the anti-parallel comes from. It's just that. It's just how they're oriented and how they're going to bind together. Because remember, a nucleotide is going to be a whole nitrogenous base with a sugar with a phosphate, repeating over and over and over again. So all of that is important. So that sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, over and over again, depending on where that carbon is at, calls it 5' to 3', but that's the backbone's the same chemically, either way. Okay. So we're going to get into semi-conservative replication. More words. Semi-conservative replication. It's not as hard as it sounds, and I hate this picture that has nothing to do with the semi-conservative aspect. It does have to do with the replication. We might come back to it in a minute. But semi-conservative replication says, if this is your DNA molecule, the reason I do it up here is because I can do different colors. This is your original DNA molecule, and we're going to go through mitosis. We're going to go this, duplicate it, and separate them out. So you're basically going from one cell to two cells, identical. This is the goal. When we're doing that, you're going to separate your strand like this. And they're still oriented, not crappy way. Okay? Okay, we separated them. Are we happy with that? We pull it apart into single strands. Now, this isn't the same as this, right? These are double strands and double strands. Now we're single stranding it. How do we get there? By this. So we're going to make copies of it. Nucleotides can only be copied by adding the 5' first. Oh man, don't you just love it? And then going to the 3' end. So that would mean starting at the 5 over here. And then this way. And it's easy enough to imagine, especially like if you have this one going like this, and then whatever, it's going to copy off of that. And now we've got two identical strands. We separated the originals and made copies of each of the originals. So each of these copies consists of one old and one new strand. Okay, that's semi-conservative because we conserve half of the old. That's why it's called that, right? Semi-conservative. So each daughter molecule will genetically be identical to the parent, and then one half of that strand is old and acted as the template, and the other one is just newly generated. Okay, so this is all done by DNA polymerase. I think it sort of gives away what it does and what it's for in the name, but it's a polymerase. ACE always means enzyme, if you haven't picked that up from any of your chemistry classes, and we'll talk a lot about enzymes in chapter 10. ACE will mean enzyme. Polymer, we're going to pick monomers and link them together, which is our nucleotides, right? And then DNA is what we're making. So DNA polymerase, making DNA. It's going to be the one that is going along this copy, this red strand, and adding the green ones in the 5' to 3' direction. So that's the enzyme that's actually going to be doing that. Right, like it says, we're only going to add them in the 5' to 3'. So it always has to be the 5' at that end. So what we're going to do is, we can't just separate our chromosomes. That's be nuts. Like that's a lot of DNA to just like separate out all of it and then just make copies. So what we do is we open it at a point. This is particularly useful to think about with bacteria because they're a big old circle. So they'll have two replication forks. We call it the origin of replications, basically. They'll start at each end and then they'll meet up. And like that makes a whole copy. So it's nice and simple. Our chromosomes are a little bit more complicated because they are in linear format. And when you're going to unwind our chromosomes, you can only unwind them to a certain point. There's waste DNA at the end of your chromosomes that will account for that. But each time that you make a copy of your DNA, you're like letting go of some of the waste because it couldn't come with you, because you couldn't get all the way to the end. That's just how it works out. That's your telomeres. That's, guys have ever heard about telomeres shortening, and that's what makes you old. If they do shorten, and at some point they can't shorten anymore. That's legit. So that's just how it goes. You can only replicate your DNA so many times. Anyways, I don't know that that is what makes you, die or anything like that, but it's a thing that happens, okay? So our replication fork is where we're opening this up, where we're gonna start this out. So we're separate those strands, we see those red strands separating out, that's gonna be kind of a bubble. And at each end of that bubble is what we call the replication fork. We're gonna look at one end of the replication fork in a minute. Because DNA polymerase can't just add on DNA nucleotides to nothing. It has to add it onto something. So it's like you can't just like stick it up here into the nothing with your nucleotides and hope that it'll stick because there's nothing to grab onto anywhere. Like if you were rock climbing, you can't just jump into the air and grab nothing, right? There has to be something there to grab onto, to build off of. So they need a primer, and that's what we call it, is primer. These are primers that exist naturally. When we do this synthetically to make copies of DNA, which we'll learn about in the next chapter, we can generate our own primers and work with that. But they can be very specific to certain things. But these guys, it'll be just RNA. It's a little bit of RNA that we can build off of. And even if you needed that sequence where you put the RNA on and you want it to still be DNA, we can take that RNA out later and then add from the DNA onto that. So that makes sense, right? So cool. So we can do that. So that's the idea of what some of our starting pieces. So now we are going to get into this replication BS. And I'm actually going to leave this and go to this next one. So we saw this picture. Let me go back to this picture over here. We saw this is a replication fork. It's like our bubble, and it's going out on each end, on each side. They're both pushing out at that point. And so these are becoming double-stranded DNA, whereas it was just opening to two blue single strands. We're just adding it on, right? So that's the idea of what's going on at the actual whole fork. This is going to be just one side of the fork, okay? Looking at it in detail. You can only add the nucleotides in which direction? 5' to 3', right? So we'll add our little nucleotides in this direction, 5', and then we're going to diddly-daddle along the way, adding our nucleotides. Thank you, DNA polymerase, for doing that, okay? Great. What about the other side, though? Because last time I checked, this is the 5', and you ain't, this is 5', I drew over it, but that's the 5'. The other side is going to be 3'. You can't add in that direction. So as you're opening up towards this fork and spreading it apart more and more as you're making the copies, what do you do with this side? So what we do is, because we have to do the 5' to 3', that's just how it goes. We will open up a little bit, and then as we get the whole segment free, add on the 5' to 3' direction. We'll do it in fragments. So imagine this opening up even further. DNA polymerase will be able to come along and then fill out that fragment. So fragment after fragment after fragment. That's how it has to go. So one strand is called the leading strand and the other strand is called the lagging strand. I'm sure you can imagine which is which. This is the leading strand and this is the lagging strand. So we create all these little fragments. Eventually, we're going to go in and seal them together with an enzyme called DNA ligase. If you don't know what ligate means to join together. So that's what it will do. So anyways, those fragments have a name that you do need to know for the exam. They are called Okazaki fragments. And I will write it, don't worry, but it is on the previous slide. This crap here is why I post the videos, okay? So definitely make use of the videos when you're getting to study time, all right? Because it'll make more sense than I can ever draw on here because they actually act it out and stuff. I can only do so much. All right, so that's it for replication. Remember, replication is only DNA to DNA. It doesn't have to do with making a protein. It's just making copies of the DNA. It's important. But yeah, so now we're going to get into the central dogma, going from DNA to RNA to protein. We've learned these terms already. We've written them on the board. Basically, all RNA that we're going to need in order to do all of this is going to come from your DNA. And the big three that you need to know are these ones. We obviously have our mRNA. That's our working copy. Single-stranded, okay? Single-stranded. These are all like basically single-stranded. This is our tRNA, which is our transfer RNA. And it transfers amino acids. We'll talk about how, don't worry. I know you're excited. Here we have the rRNA. This is ribosomal. You can also call rRNA regulatory RNA, but that's a different thing. But anyways, for our purposes, we only care about the ribosomal one. Ribosomal RNA is what makes up the structure of the actual ribosome. That's going to be the machinery where all this is going to happen, right? We already talked about the phenotype versus the genotype. Your gene is the blueprint. We're going to make our copies of. So when you have DNA, and you look at it this way, we will divide it in 3 nucleotides. So imagine that this was a G, T. I can't read the T, but you know what I'm trying to say, okay? This is called a triplet in the DNA. And they're always going to be in 3s. It's very important, that part, okay? Whereas when we make our copy, our working copy with our mRNA, our messenger RNA, These are called codons. Now, if you look at the structure of the mRNA that we have here, just literally based on the shapes that it has in the picture, you will see that that structure of our mRNA looks exactly like the bottom strand of our DNA. Right, that's where we're getting it from. But did we actually make it from the bottom strand? No, we couldn't have, right? We had to get the complement in order to make it. So it just happens to look like it. So the one that we're using as a template, go figure, we're going to call it the template strand. And the one that looks like it is the coding strand, because it's what actually would carry the code if we were reading it directly. Okay, so template strand, coding strand. The template strand is literally what we're using as the template to make the mRNA so that it looks the same as the coding strand. So now it does. So we have it separated into 3 nucleotides, A, T, A, G, U, or whatever in this case, or the complement of it in this case, because we don't use T in mRNA. So now we've got our mRNA, and we can take these codons in the triplet form and make an amino acid. So those 3 nucleotides on the mRNA that we call codon code for 1 amino acid. And that code that we're reading out on, the ACC, the UAG, the whatever, going to spell out your protein with the corresponding amino acids. There are 20 amino acids, and these guys are going to basically cover those. Okay, so a lot of different variations in your protein coming from this. That's what your genes are for. That's what they do. All right, so here's all of our players, our major participants. We kind of already went over these. If you'll look at our transfer RNA versus our messenger RNA. So our messenger is down here, and our transfer, it looks like an upside down T, kind of, right? We can see that. Transfer RNA, look at our messenger RNA codon, AUG. I keep writing on it. AUG. A will bind with U because it's replacing the T. U will bind with A, and then G binds with C. You guys see how that is complementary to it? These guys are going to come along and see your single-stranded mRNA and pick it up with their complementary sequence. And they're carrying an amino acid. Starting to see how this is going to fit together? Okay, so the ribosome is going to do all this for us. God, it's nuts, but once you start seeing it, it makes a little bit more sense. Okay, so we already know about RNA. It uses ribose. It uses uracil instead of thymine. It's the same otherwise, though. And it will still have a helix, even though it's single-stranded. Okay, our messenger RNA is our working copy. Then we have the codons, that these came from the DNA triplets that are going to basically code for the amino acids individually. The transfer RNA will meet up with it. Its structure that is complementary is called the anticodon. Not crazy, right? So our codons on our mRNA, codon. Each codon is going to match up with a anticodon on a tRNA, always in triplets. Okay. I wish the videos I could put them in here because it would make more sense, but it won't let me actually embed them. It like says that it's not like accessible or something like that. So whatever. But do go watch them, please, because it's way better than I can explain it. So ribosomal RNA is what's going to make up our ribosome. I don't need you to know too much about these ribosomes. I don't need you to know the size. I just need you to know that it's going to be a piece coming up with another piece, clamping on to our messenger RNA. Just like imagine you're just holding on to it. All right, so it's going to be important when we start into translation. But of course, why would we do that? Why would we go in order of everything? We're going to go back to transcription now. Because before we can translate anything, how did you get your mRNA? Exactly how you would imagine it. You're going to open up your DNA like you did for replication, but now we're using RNA nucleotides. And how does it decide that? The RNA polymerase. So it's a whole other polymerase that will only work with RNA nucleotides, right? Anyways, so our first step in this is going to be called initiation. It's whenever your RNA polymerase binds at a place on the DNA called the promoter sequence. When the RNA polymerase binds on the promoter sequence, we'll also have separation of the DNA. So we can start sticking some RNA nucleotides on there. Guess what this guy does? it also only operates in the 5' to 3' direction. So that's what I'm saying. Like, I'm never gonna, y'all, it's always gonna be that way. So adding our nucleotides in the 5' to 3' direction, as you can see here, 5' is what we're spitting out first. Adding our nucleotides, add our nucleotides, add our nucleotides based on, you know, if you're the mRNA, you're gonna add the A if you see a T on the DNA, but if you see an A on the DNA, you add a U, right? That's important to remember. And then G to C, it's all the same there. Just keep adding on as we're cranking on down until you get to a point that it basically tells the RNA polymerase to knock off. Just don't bind on anymore, just take it off. And then you're done making your mRNA transcript. We call it a transcript. Like a script, like you would read. So that's it. There's 3 steps: initiation, elongation, and termination. And it makes sense. I feel like this one makes sense. Let's see where we are. 6:30. I don't know how I feel about it. Yeah, we're going to do questions. We're going to do it. This is hard stuff. You may use your notes. Please feel free to use your notes because this one is ridiculous. I'm not under any impression that it isn't, okay? All right, so our first question is, what are the 3 parts of a DNA nucleotide? Taylor. You're okay. Yeah, yes. Nitrogenous base, yes. You're okay. I know you guys can't say some of this stuff. Like I've said before, if you guys can't pronounce a thing, I'm not going to hold it against you. I just want you to know it when you see it on the test or in your notes or whatever. All right, so good job. So yes, it's going to be the sugar and the phosphate and the nitrogenous base, in case you guys didn't hear. All right, what makes DNA replication semi-conservative? All right, Haley. Yes, so one old and one new. That's exactly right. We split it apart. So consider it old and old. Split it, make new ones on each side. So it's old and new, semi-conservative. All right. During DNA replication, which strand, what's the name for it, makes the Okazaki fragments? We have Analia. Okay, sorry. So this one, right, so the short segments, that's exactly right. So the one with the short segments, they're going to be made 5' to 3', so you're absolutely right with that. The name for those, for the one that makes those fragments is going to be lagging strand, right? So it's the name of the strand. Yes. So that's good, though. You had the concepts there. Yes. Yes. So yes. So whenever we make them, like I was drawing up here, like this is 5. It's hard to tell it when I write it up here, and I'm aware of that, but I can only write so well. You guys see my handwriting. So, but yeah, let's just put the 5s up. That'll help. Okay, that's all the 5s. All right, so 5' to the 3', 5' to the 3'. So when you split them apart, I kept them oriented that same way so that you would know, okay, this one's 5 to 3 on the top, and so the 5 to 3 has to go that way. This one's 5 to 3 this way, so that one has to go that way. Does that make sense? Yes. And it'll, like I've said, like the videos just make a little bit more sense than I can draw it. But all right, good question. All right. Generally speaking, what is the overall purpose of transcription, Jenna? That's your luck, right? For asking the question. All right, the purpose, the general goal of it. Yeah, that's it. So making that RNA, if we're transcribing, it could be messenger RNA or any of the other RNAs as well. It's all still called transcription, okay? All right, what is the function of messenger RNA? I have Christy. Yeah, it's working copy. Yeah, so it's the working copy of the gene that you're wanting to make the product of. So good job, guys. That's the last question of this one. You can sit down. Thank you for putting up with it for once. It's been a long time since we were subjected to this question. Okay, so moving on to making our product. Our messenger RNA, again, we're going to read it in codons, those groups of 3, and those codons are going to dictate which amino acid. This is a chart that I can't tell you how much I hate. When I was in biochem, like I said, I didn't know all of these. I didn't know if you told me UCA, I had to tell you that it was serine. I didn't know the abbreviation, which isn't even up here, for serine, and I had to be able to draw you serine. That's what we had to learn how to do. I'm not going to do that now because I can't. So you're welcome. But back then I did. Anyways, so we've got all of our 20 possible amino acids. And if you look at these, let's look at phenylalanine because it's up here at the top. And most people have heard of phenylalanine before, like phenylketonuretix. There's phenylalanine in there, and their inability to process it. But anyways, phenylalanine, it's coded for by your mRNA saying UUU or UUC. We've got 2 there that'll code for that. Whereas serine, like I just brought up, which is here, can be coded for by any of these. They're all UC anything. So some of these can be coded for by many codons. Now, why would that be? Because look at all the different combinations you can have, guys. It's more than you probably thought. When I told you like all you have are ATCG or AUCG, you probably never thought that it would be this many combinations. These are all different combinations and only 20 amino acids, but it is enough. 20 amino acids is enough to make you and everything that ever made anything in you. It's pretty crazy to think about. But these are all of them. Some of them you have to be supplied with these guys, but it will still be coded for, and a tRNA will be assigned to it, just you can't make it in your own cells. Interesting, like lysine. But anyways, so there's lysine in case you were wondering. You guys, does anybody in here know what I'm talking about when I talk about the lysine contingency in Jurassic Park? Anybody even know what I'm talking about? Okay, like we get some like subtle nods, like we don't want to admit that we're dorks. But yes, that was a thing in the movie. Anyway, so it's true, though. It's the truth. All animals have to be supplied this amino acid. And it's something that can be found in plants. And because if you're a plant, you know, a cow might eat the plant, and then we would eat the cow, or you know, we could eat the plant too. But you could also get it from eating the cow if you're a carnivore, for example. So I get it from other ways, but yeah, interesting. So the reason we have all this overlap, why you have 4 UC anything is serine. The reason you have that is because of mutations. So if you had a mutation that changed your UCU into UCC, it would still code for serine and it wouldn't mess up your protein. So that's basically where this is going to come from. There are a few that are special on here. AUG is always going to be the start. It's always going to code for some form of methionine. Always. We don't always keep it on there. You can always clip it off later on, but it's always going to be the first one. And then we have 3 of them over here. These 2, UAA, UAG, and then UGA. These are our stop codons. They're stop. They do not carry any amino acid or anything on there. They won't even call for a tRNA. In fact, because of that is why your ribosome knows to dissociate and not be there anymore and stop. So cool. So here's how it goes. Here's back to our triplets. And remember, our template strand here and our coding strand here. So if it's our coding strand, it should read basically the same thing as our RNA. ATG on our code would be AUG down here. So that's the only difference is those U's. And we use the template strand to make it. Great. We see our codons separated out there. The top is the DNA triplets. Next, we have the mRNA codons. Next, we have the tRNAs that are going to match up AUG, A to U, U to A, G to C. These are our anticodons. Codons to anticodons. And these guys are attached to an amino acid. So because that said AUG, like I said, it's always going to start with AUG. AUG saw the UAC on the anticodon. That binds on methionine that was attached to that tRNA to our growing protein. Okay? Or in this case, starting our protein. So there's some pictures that we're going to look at that I feel like make a little bit more sense. But there are 3 stages to this as well, and it happens to be the same as the transcription ones, okay? We'll have initiation, elongation, and termination, same concept here. It's just we're talking about it for translation now. So we're always starting with AUG. We have our various stop codons that we consider to be nonsense because they do not code for an amino acid. And then translocation is the term that we use for when we shift our ribosome down. So that's keep moving down, right? So here's pictures. There's a video of this. So your ribosome comes and binds onto your little yellow strand here. That is, let's zoom in. I think I can. I just have to figure out how to. Okay. Okay, I think it's yes, this is good. Okay, so I definitely need it on this one. I'm not going to read you those whole boxes. If you want to read the boxes, you're welcome to. But basically what's happening here is your little yellow mRNA with its codons, just like we saw in the previous slide, are matching up with their anticodons on the tRNA that is attached to the amino acid, just like we said. So it happens in these little slots in the ribosome. So we've got the first slot with our starter. The next one will have our next match go in with that amino acid. Then we're going to create a peptide bond with that. So that's just, a peptide bond is just what we do to link our amino acids together, okay? When that happens, the first one that we had on here originally doesn't need to be there anymore. So it will detach from its amino acid and it'll just kick out. The tRNA is empty now, and it'll just kick out. So then guess what happens? This spot is empty, right? So this guy, shift. The next guy, come in. And it will keep doing that over and over and over and over again until we get to a stop. And just keep sliding off, kicking out the empty one, sliding over, making our peptide bond, kicking out the empty one. And eventually you're just left with all this 1, 2, 3, 4, 5, these purple squares, essentially. Our amino acids linked together. That's the best I got. The video is a little bit better. Oh, I can move this around. Jeez, look at this, y'all. What have I been doing this whole time to you? Okay. But yeah, here we go. Our empty tRNA getting kicked out. It's the exit spot. That's what the E is. Open up that first P site, which is like the primary site. Shifting that over, which is on the next slide. How do I get out of it now? Oh, here. Okay. Yes, so then we'll shift over. The next one can come in and so on and so forth. You guys get it. So, okay, so when you are a bacteria, you can do this faster than eukaryotes can by far. You remember how we keep our DNA in our nucleus. And so we have to go into the Smithsonian, make the copy of our mRNA, take it out to the ER where the ribosomes are, and then make our translation product, our protein, right? We have to take it out. They don't, because they don't have a nucleus, right? So they can just do it all right there. And by saying that, they can just separate their DNA, start making their mRNA, and as it's being still made, we can attach on our ribosomes and start making the protein before we even finish making the messenger RNA, which we can't do because we have to get that messenger RNA made fully first. So pretty cool. We call this co-transcriptional translation. Yes, isn't it nice? Because while we're doing transcription, we can start translating. Okay. They can get all this, look at all these ribosomes. They can get lined up, just taking out protein while reading along while we're still making it from the DNA. That is just crazy. And then get pictures of that. All right, so this is the COVID mRNA vaccines. These are the vaccines that I told you guys about the guy, right? I told you about the guy that I did work with. Okay, this is the work that he did. His stuff was this. It just happens to be in your textbook also. But he designed these mRNA vesicles that were just wrapped up in like lipid bilayer, and inject it into your arm, and your cells will just start translating that mRNA, and then they'll make the protein, which happens to be the spike proteins of COVID, which is why you get the rash down the arm and stuff like that, because your body is like, your arm looks like not right. But it's what we could get our bodies to do that quickly in that amount of time while people were dying. Like we said, this is not perfect, right? We know it doesn't protect completely. It's not like measles vaccine, where that can protect you pretty much like forever with one vaccine, and you wouldn't even know. It works so well, or like to remind people of it. It works so well, your measles vaccine, that you can go to the fair or to the mall or to a concert or anything. If there was anybody there or any number of people there that had the measles, you would never know because you would never get any symptoms. Why? Because you never got the virus in you? No, you got the virus in you. Your body was just ready when it saw it. It's that ready. So that's crazy that your body can be that ready. That's immunology. But this, we had it good enough. Yeah, you still get COVID, but now you feel like you have the flu or a cold for a little while instead of being, holed up in a hospital and potentially dying. Otherwise, healthy people were dying from that. So let's not forget it. I hope so. Yeah, so that's the goal overall that I feel like everybody wants to strive for. It's just some proteins are just harder to get the body to respond to effectively. So we can't really choose that. And that's I think, I don't want to get into conspiracy theories or anything, but it could potentially be an artifact of the fact that they probably work. Please don't try to argue with me on this because I absolutely believe this wholeheartedly, and I think you probably should too. But that China was developing a bioterrorism weapon that got out earlier than they expected for it to. And if you don't believe me, look at what they did with it after, because they still worked on it after that. And now they have a version that kills humanized mice, which means these are mice that have sensitive human proteins genetically implanted in them. This is facts. This isn't conspiracy. This is actually like real facts. This one killed them 100%. You think that wasn't a bioterrorism weapon? I don't know what to tell you, but yeah, that absolutely was what it was. So that's me. That's my conspiracy theory of the day. I don't know. So here we go. We can't do that at the Simply Reset about that. We have an inability to do that. We have to bring it out of there. The other thing that is very, very different in how these things operate, we have introns and exons in eukaryotic cells. So this is us. So of course, we're more complicated in general. Our cells are anyways. We have more chromosomes, so that's complicated. This is another way we're gonna take it up a notch with complications, okay? Introns and exons. Introns are sequences that you don't need in your mRNA. They could potentially be needed if you got the right signals, right? But if you're making this particular protein and you don't need them, we call them introns. If you're making this particular protein and you do need it, we call it exons. So, great example, good one that I just basically sent you guys just now. There's all this information in the notes that I'm trying to teach you guys that's important. Consider that the exons. The introns are things where I tell you about conspiracy theories and whatever you don't really need it. So you can just cut that out. You get what I'm saying? So that's how these work too. Cutting out stuff you didn't need and joining together stuff you do need. This happens in a structure called a spliceosome. This is called splicing. Your cells do this naturally all the time. So what you do is you make your mRNA, but instead of you just like straight up carrying it out of the nucleus, first you have to edit it and cut out the scrap. That's really what happens. Bacteria don't have this. That creates a problem when we get into genetic engineering, which we'll talk about in the next chapter. I don't want to get into it now. So introns and exons, very important. Spliceosomes are what does it. Okay, now let's talk about operons. I don't know. So operons are basically a collection. I don't like them because I feel like they come off so complicated, especially after we talk about all this complicated stuff, but they're really not meant to be. So hang in there. Operons. They're only in bacteria. The archaea do too, but who cares? So bacteria do this. We do not do this. Operons is basically like a set of genes that are associated with a task, but you need multiple proteins to do the task. You can just make that long mRNA, all of that, and then take out all those proteins from that one mRNA. We have to make a single mRNA for every protein, but they can have a whole bunch linked onto that one mRNA. That's an operon. Okay, so there's two kinds of operons. They don't just exist naturally like that. They are either inducible or repressible. If you can be induced, if you need to be induced, and if you want to think about it as inducing labor, you can, because a lot of that makes sense to a lot of people. If you need to induce somebody to go into labor, Why would that be? Because they are not going into labor, right? They're not doing it. So think of their labor is turned off. You're going to have to induce it to turn on. Okay. So repressible, this would be, I don't know, if you were trying to keep the labor from going so fast, I don't know, like it's going too fast, naturally on its own, the patient comes in and you need to slow it down, so you want to repress it. Well, you can't do that unless it's going, right? So it's going to be on already. So you're not going to repress something unless it's already on. You're not going to induce something unless it's off first. Okay. Cool. So here's our inducible operons. We have genes turned off, and they're going to be turned on or induced, usually by a nutrient. So some nutrient maybe you don't see very often as bacteria, like lactose, which is the example it's going to be, by the way. You come see this sugar, lactose. We get it in our milk, right? Same old lactose, not any different. But the bacteria don't usually see it. So now they're seeing it. So they'll turn on this operon so that they can deal with the lactose, okay? The lactose is what's going to turn it on. I'm going to show you how. We're going to get past this for a second. All right. Oh gosh, for crying out loud. Okay, this is the picture of how this would work. You guys remember how I said your RNA polymerase is going to bind at your promoter? Like when we were making our mRNAs? Yeah, so it still does. That's not magic or anything. We can have right next to it in that orangey brown area where that little horseshoe thing is binding, that is the operator. So it's right next to the promoter. And we get the horseshoe thing, which is called our repressor protein. It'll be sitting there naturally, okay? In this case, remember they're turned off first in order to be induced. So it's turned off because the horseshoe is here. We can introduce our inducer, which happens to be lactose. It binds with that repressor protein, and we know how proteins do when they bind with things. We have these new bonds happening, so they often will fold, change their shape a little bit. That's what happens. Our horseshoe kind of opens up almost like a clamp, like a clothespin, and it'll come off of that operator area. So now RNA polymerase will start reading on down. It can be fine now. When lactose is present, causes the repressor protein change shape and come off, and now it's been induced, right? No. We'll make our enzymes to break down and digest our lactose. And eventually, when there's no lactose left to work with, we won't need it attached to the repressor anymore, and it'll go back and bind back on and go back to how it was. Okay, so that is the inducible one. It has to be turned on, which means it was off, which means there's something preventing it from doing its thing. Okay, this is the repressible one. These guys are on, and now we need to turn it off. One example that we have here is the arginine one. These guys are going to be making proteins relating to generating arginine. It's an amino acid. Okay, this amino acid, we need it. We're making more proteins. We're building stuff in the cell. We need the arginine, and the cell's using it up regularly. And it's not building up or backing up or anything. Well, it's a supply and demand thing. If the cell doesn't need the arginine anymore, then we have a buildup. And when it builds up, now it will bind with the repressor because there's enough floating around in the cell, whereas before it was getting used up. So bind with that repressor, it changes its shape so it binds on. And that's what represses the genes until we need more arginine, until we have used it all up. Okay? They really are not too complicated. Inducible operons can be turned on because they were off. Usually something we introduce that is causing them to be turned on, inducing them. Repressible operons are just on, and then they're going to be turned off. Okay. Recombinant, basically anytime we have recombination, this is when any organism contains and expresses genes contained in another organism. They don't want to be different species. We like different kinds of bacteria or sharing like antibiotic resistance. Okay, that's recombinant. That's a recombinant. Usually we see this occurring with horizontal gene transfer in bacteria. Horizontal gene transfer is vertical is whenever you're going to transmit from parent to child. So like if you're a bacteria, if you're dividing and you're spreading your genes on to your children, that would be vertical. Horizontal is like if I come up to you and we hook our little like sex pili together, like we said with the cells, and ship our little genes through that channel that we have. Okay, so there's 3 different ways that we can do that. A lot of them can involve plasmids. Remember, those are extra bits of DNA that are circles. They have genes that are not necessary for survival, but can have useful traits like antibiotic resistance. So the 3 different ways that you can do this are conjugation, transformation, and transduction. Conjugation, that was the sex pili, where we create the channel between the 2 bacteria and it can shuttle DNA across. That's why it's called sex, so it's called conjugation. All right, transformation, we have a breakdown of DNA, usually from dead cells that are in the area, and the cells around it can pick it up and incorporate it into their genome. And then transduction is whenever you're having new genes introduced into you from a virus. This is where we get that lysogenic conversion from. Cool, cool, we already did it, we did that. Our mutations, these are any change to the nucleotide sequence. Obviously, this is a driving force of evolution. We'll talk eventually about, antibiotic resistance. These can be spontaneous. They had to come from somewhere, right? It didn't always get traded around. Spontaneous mutations versus induced mutations. Spontaneous mutations are just going to occur naturally from errors in the genetic code. There's a video somewhere on one of my talks that's posted on Canvas somewhere. But it's like a whole auger table, and they put E. coli at the ends, and it grew on there fine, it's just normal auger, and it filled up that whole area. Next to it was a normal concentration of antibiotic, and it was 10 times, and it was 100 times, and it was 1,000, and went up to 10,000 in the middle. And just because of spontaneous mutations, they didn't introduce anything that's normal E. coli, just like we would use in the lab. Not anything crazy, just normal. It developed a mutation spontaneously and grew to the next area. And of course, that fills up, takes another mutation to make it through the next one, and went all the way to the middle within a week. So that just happens naturally. And then imagine that they can spread it to each other. So you can see why we need to take our antibiotics all the way to be sure we're killing them all and not letting that happen, because that'd be nuts. That's the idea. All right. Cool. Induced mutations, obviously that's going to be things causing the mutations. This would include things like UV radiation. When you go out into the sun, it causes UV damage to your actual DNA. And your cells can repair that, but some organisms can't. So that can lead to mutations that can become permanent and heritable potentially if you're a bacteria. So the different kinds of mutations you can have: missense, nonsense, silent, and back are the normal going to be like traditional nucleotide to nucleotide changes. All right. So a missense mutation. Let's say U was phenylalanine, and it got changed. I don't know what the actual letters are, so please don't hold against me. I didn't remember. C, and let's say that is leucine. I don't know. Well, phenylalanine and leucine are different. We only change one nucleotide, and they could even be wildly different. If we were talking about something that was, hydrophobic, changing to something that has a charge, that could make a huge difference in your protein structure. But that's a missense mutation. We have a new amino acid. A nonsense mutation, just like we called the stop codons nonsense, that's what we're talking about, stop codons. They don't any longer code for any amino acid. They get changed to one of those stop codons. So C to U, I don't know, UG, I don't know what they are, but you know what I mean. Changing it to the stop codon. How long it would take was be one change for some of them. Silent mutation, please. The silent mutation, this is going to be, if you went from U to A, and let's say that these are both phenylalanine, who cares, right? Your protein doesn't care because you're still coding for the same amino acid. So that's silent mutation. We have the same amino acid, even though we do have a change in the nucleotide sequence. A back mutation is like if you had changed to UUC, and then your next generation went back to UUU. That's a back mutation. So those are the basic ones. This is just showing some of them out. I kind of already walked us through. You're welcome to look at some other examples. We just don't really have time right now. Frame shift mutations. These are going to occur whenever you delete or add a base or two. What do I mean by that? Imagine you had a book and it's Harry Potter or whatever you want. Harry Potter, and like on the third page, you delete one letter. Okay. keeping the words the same length, shifting every other letter afterwards into that spot. You could see how that would screw up like the whole book. That's exactly what a frame shift mutation does. So we have a lot of starts and stops in our genetics. It shouldn't affect necessarily genes downstream, but it could potentially, but definitely will screw up that entire protein that you were supposed to make and replace a whole ton of amino acids and ruin it. So frame shift mutations are very dangerous. Deleting one or two. If it's in triplets, then we're still gonna be okay, kind of. But yeah. All right, the last thing is the Ames test. This is a test that we do to check for mutagen potentiality. So any mutagen or carcinogen, these are things that are going to cause changes in your genome. So we can take salmonella as an example that they give us. We can take salmonella and delete its ability to make histidine, which is an amino acid. Okay, so it has to be supplied. If we were taking out the lysine contingency, they wanted to pretend like that was a helpful thing. Take away that ability to make histidine. It shouldn't be able to grow if we don't supply it with histidine. We expose it to our mutagen or our carcinogen, whatever that chemical could be, UV radiation, whatever it is. And then I see if it'll grow. And if it grows, right? That'd be a big deal. We don't want that. So that's how we look at carcinogenic potential of things. Carcinogens tend to come from mutagens. You're altering your DNA, and sometimes that can affect your cell cycle and cause it to grow out of control. So that's that. I think that's everything. We'll go up to the lab.